Okay, well, yeah, I want to thank Dennis for inviting us to present this paper that we just posted on the archive a couple days ago. As you mentioned, this was a little bit last minute, so I apologize if the slides are a little rough or my presentation is unrehearsed because I haven't rehearsed. But yeah, I'm glad to walk you guys through the paper today. The title is The Medical Relevance of Protein-Truncating Variants Across 337,208 Individuals in the UK Biobank Study. And as I mentioned, it's available on bioRxiv. So just as a quick outline of what I'm going to present today, I'm going to start with some background on protein truncating variants, or PTVs. Then I'm going to go through the results of the paper, which we've kind of split up into three areas, sort of a GWAS, which was our initial analysis, PheWAS that was focused on a subset of genes of interest, and then sort of a collapsed analysis where we collapsed the genotypes of PTVs in a given gene and did an additivity analysis. And then finally, I'll summarize the paper and provide just a couple areas that we're thinking about moving towards. So let's go ahead and start with the background. So protein truncating variants, or PTVs, are genetic variants that are predicted to shorten the coding sequence of genes. So, it's important to recognize here that these often are thought of as loss of function, but they can also be gain of function. They can just cause early termination of transcription or translation, but they may still lead to a functional protein. It just might not function the same way that it normally does. But often people do think of these as loss of function. In the image here, we have four common ways that PTVs can shorten the protein coding sequence of a gene. In panel A, you can see you could have a nonsense SNP that causes an early stop. In B, you could have a frame shift indel that shifts the frame of translation of the protein. C could be a large structural variant that deletes an exon or something important and also causes perhaps early termination of transcription. And in D, you have splice disrupting variants. For this paper, we focused on A, B, and D because using the UK Biobank data with the array data that was available, we don't really have information on large structural variants. So why are we interested in PTVs as a class of genetic variants? Well, if PTVs disrupt protein sequence, then you kind of have this prior sort of hypothesis that they will have a large effect. They could have a large effect on phenotypes of interest. The other thing that's really nice about a PTV is if you see an association between a PTV and a phenotype, you may feel like this offers some amount of strong evidence that the gene is relevant to that particular phenotype, unlike maybe a non-coding association where you say, well, now we need to fine map this, we need to figure out which gene it's targeting. Here, if you're knocking out the function or dramatically altering the function of the gene, we think that this offers strong evidence that that's the actual gene that's important, although that's not always true, and I'll show that in a minute. The other thing that excites us about PTVs, and I think a lot of people are thinking about right now, is that we think that they offer a good target for drugs. So, if you're walking around with a PTV that disrupts the function of a gene and happens to offer you protection against some disease, then this sort of gives you both an in vivo validation of safety and efficacy, right? These people are walking around with a PTV without the function of some gene, and they're okay, but they're protected against some disease. Or maybe they have slightly increased risk for some diseases, but decreased risk for others that they want to be, that they don't want to get. And so I'll show that a little bit later too. So PTVs have been implicated in a variety of diseases. I think the best known example is PCSK9, where I think a few different PTVs can lower LDL cholesterol dramatically in the people that have them and protect against heart disease. And this was discovered a decade ago. And recently, of course, has been shown to be clinically relevant in this paper that came out earlier this year, where you actually do see decreased adverse cardiac events when people use a PCSK9 inhibitor. So this isn't the only example that we have of PTVs that protect against disease, though. I have this little table down here that shows some of the other genes that have been involved or have been identified that offer protection against different diseases. And I apologize that I don't have the references on here, But we did write a little blog post if you want to check out the Revis Lab blog that has all the references. And these are, I believe, all referenced in the paper as well. I'm going to come back to this in a bit. So as far as the UK Biobank and why why we did this study in the UK Biobank and what it offers us. So for those that aren't familiar, the Biobank is a prospective cohort study on about 500,000 people in the United Kingdom. There's extensive phenotyping via person-like questionnaires, also hospital inpatient records and diagnostic exams for some things. They also perform genotyping using a custom-designed genotyping array with about 800,000 variants. And so this is really useful for thinking about PTV and identifying PTV associations, because there was a large number of PTVs that were included on the array and are directly genotyped, which is nice. We don't have to worry about imputation. And in particular, we might be worried about imputation because many PTVs are rare. So you need both direct genotyping and a lot of subjects to be able to identify some of these associations. So the UK Biobank is probably really the first opportunity we've had where we have a lot of phenotypes, a lot of individuals, and direct genotyping of a large number of PTVs, which allows us to really kind of get at what are PTVs doing across a broad range of phenotypes. So the kind of basic question, as I guess I just kind of said, is that we're trying to answer in this paper is, are polymorphic PTVs associated with medical phenotypes, or which ones are they? By medical phenotypes, I mean, we could be looking at, we looked at things like diseases, but we also looked at things which may not be a disease per se, but are of interest medically, like high cholesterol or something like that. And just as an overview, so I'll come back to this figure at the end and it'll be filled in with more information, but this is kind of the basic layout of our study. So we used 337,000 individuals from the UK. I'm going to go through where that number comes from. We defined phenotypes using the hospital inpatient data, the verbal questionnaire data, and the cancer and death registry data, and I'll go through a little bit about that. As I mentioned at the beginning of the talk, we are interested in PTVs, and these are the three classes of PTVs that we're really focusing on here, and I'll talk a bit about the annotation. And then this is the sort of, on the right, you can see the sort of progression of analyses that we performed. Great. So let's go ahead and jump into the GWAS analysis, and I'm going to go through some of the methodology first and then present the results. So for the subjects, the latest release of the biobank data, which was about 3 or 4 weeks ago, I guess now, contained about 488,000 individuals. But for the purpose of this study, we really wanted to get a nice clean set of individuals. So what we did was we only kept subjects that met the criteria on the slide here. So basically self-reported white British ancestry, used to compute principal components. So this was something the UK Biobank did. Not marked as outliers for heterozygosity or missing rates for genotypes, do not show putative sex chromosome aneuploidies, although I will say that we only focused on the autosomes here, and have at most 10 putative third-degree relatives. So in the end, that's how we got from the sort of 488,000 to 337,000. And I just wanted to mention this. I mean, it's in the methods, but I think there's definitely been some talk on the UK Biobank mailing list on what's the best way to subset these individuals if you want to get sort of a homogeneous group of people. And so this is the way that we went about doing it. As far as the PTV annotation and filtering, we identified about 27,000 putative PTVs on the UK Biobank array using the Lofty plugin for VEP. I apologize, I don't have the URL here, but you can find it online. It's from the MacArthur Lab. And these are putative because they're just computational predictions, although from here on out, I will refer to them as PTVs, but there is always, of course, the chance that they may not be a PTV actually, or that they are a PTV for a transcript that maybe isn't the most important one. I mean, these are issues that I think we all deal with, but that's something to keep in mind. From this 27,000, we filtered out PTVs that met at least one of these criteria that you see on the slide. So there's a genotype missing this rate greater than 1%. We also checked the minor allele frequency of the PTV against the NOMAD NFE estimates. And if we saw that there was a big difference, we sort of removed the PTV. We also looked to see whether it was multi-allelic in NOMAD in a way that sort of looked like it was making the UK Biobank calls inaccurate. And we also looked by hand at a number to determine whether there was sort of bad cluster plots. So, this GWAS was sort of an iterative process where we would we'd get some number of hits, and we'd go and we look at them, and then we'd go and we'd look, especially for everything that was significant, we'd say, okay, well, let's pull up the cluster plots and see. And there were certainly some variants that you could tell by looking at the clustering data from the UK Biobank were just not accurately genotyped. And so after applying all these filters, we were left with about 18,700 PTVs, of which 18,228 were polymorphic in the UK Biobank population that we looked at here, these 337,000 individuals. And once again, I apologize, I should have the URL on here, but we've released some of this variant filtering information on GitHub. If you look up the Revis Lab GitHub, or I'm glad to provide any of this information afterward as well. We've released variant filtering information just for the variants that are on the array from the UK Biobank. Some of it, in particular here, we've provided some sort of manual annotations for the PTVs, so we think it's a pretty good set and something we'll continue to update. We've also just provided some statistics for some of the other variants, so hopefully that's useful for people. Okay, so the last thing before we get to the results, let's take a look at the phenotype definitions. This was an important part, and it's not actually something I did. Greg McInnes and Adam Lavertu, who were rotating with Manny, did this during their rotation, and they did a great job, though. So basically, for all the phenotypes that we're looking at, they were defined by combining questionnaire responses with ICD-10 codes from the hospital inpatient records. So over on the right, these are just a subset of some of the medical phenotypes, if you will, that we're looking at. On the top, you can see some of the cancers, and the blue indicates the number of cases that come from sort of in this case, the cancer registry, and orange is how many additional cases we got through self-reported data. And you can see that for some cancers, they're well represented in the registry, but for others, like the male genital tract cancer, or cervical cancer, for some reason they don't show up in the registry. Maybe they're called something else, or maybe they just don't track it. But in any fashion, these are things we wouldn't have been able to even look at without including the self-reported data. And certainly you can see below in panel B, these are some of, and this is just a subset of the phenotypes we looked at. You can see some of the other things like depression or osteoarthritis. These are just entirely self-reported, so based on the questionnaire data. I think there was just a paper that was just posted to the archive within the last day or two, looking at sort of phenotype definitions in the biobank and showing that the questionnaire data really seems to be actually quite useful and not as messy as maybe some people feared. And that seemed to be our experience as well, that the questionnaire data is really useful. And I think you'll see more from us on that particular question in the near future, hopefully. So in the end, we were able to define 135 phenotypes that had more than 2,000 cases. And that's what I'm going to focus on for the first part of the talk. And then later on, I'll bring in a few more phenotypes. So we'll drop down to 1,000 cases. I guess I should mention here, too, that we didn't look at any of the available. We just decided to focus on these sort of binary medical phenotypes. But we'll see more on quantitative traits in the future. And I'm sure a lot of you have seen, but we've been sort of posting these results on our global biobank engine. The URL is here on the page. So if you're interested in looking at these results yourself, besides what we've reported in the paper, I'd recommend checking out the website. And I believe the quantitative traits are actually uploaded there now as well.
Great. So for the GWAS, the way that we did this.
Chris,
yeah, please.
Quick question from our side.
Please.
I'm just curious for the ICD-10 codes, did you tend to truncate at 2 digits, or was it sort of a mixture of 2 and 3 digit codes to define your phenotypes?
Yeah, I believe it's a mixture of 2 and 3 digit codes. As I mentioned, I wasn't the one that did that, so I honestly I was a little bit of a black box. But yeah, I believe that it's a mixture. So they kind of went and they sort of descended the tree and kind of pruned it a little bit to get the things that were useful.
Thanks.
Yeah, absolutely. Glad to answer any questions anytime. Yeah, so for the GWAS, the way that we did this, and I don't know, maybe this is better called a PheWAS, to be honest, but who knows at this point. So what we've done is we took the 18,000 or so PTVs that we identified, and we decided to really just focus on the ones that had at least a minor allele frequency of 0.01%. So that's kind of what we decided to focus on for this. And we split them up into 3 bins. And you can see the number of PTVs that fall into each bin here of minor allele frequency. So there are a lot that are down in that lower bin, and there are still a lot more that are even more rare that we aren't looking at here. There's about 700 that have a minor allele frequency between 0.1 and 1%. And there's 463 that are between 1 and 50%. And so we ran GWAS for these variants against these 135 phenotypes that I mentioned that had at least 2,000 cases. And we used Plink2 to do this, which is an updated version of Plink that Chris Chang has been working on. And it's quite fast, and it's really useful. In fact, when we ran these GWAS, we ran it for the entire UK Biobank array, but I'm just focusing here on the results from the PTVs. And then the reason that we split them out into these bins is we sort of applied the p-value adjustments separately for each bin. And that sort of just reflects the prior knowledge that we have that you expect sort of different levels of power, of course, for these different minor allele frequency bins. And you'll see that reflected right now if we go and take a look at the Manhattan plots. So overall, under this criteria, we identified 75 associations that had an adjusted p-value of less than 0.05. And 25 of these are outside the MHC. So here there are 3 Manhattan plots, 1 for each of the minor allele frequency bins that I just showed you on the previous slide. And you can see that for the more common bin, the 1 to 50% minor allele frequency, there are a lot of associations up there, but especially there's a lot in the MHC. So the region where the scatter points are small, those are all PTVs in genes that are in or near the MHC. We kind of have a little bit of an extended MHC definition here. You can see that in the middle bin, we have some other associations, including some with genes that are in or near the MHC. And then down in the lower bin, we really only pulled out 4, which is maybe not too surprising. These are still pretty rare variants. Okay, so just to dive in a little bit to some of the associations that we found that we thought were kind of interesting. So we identified some that were very medically relevant, or sort of, I guess maybe we know about medically. So TSHR, for instance, is the thyroid stimulating hormone receptor. And we found that if you have a PTV and TSHR, then you are at risk for hypothyroidism. And that makes sense, right? If you sort of lose the function of the stimulating receptor, then your thyroid doesn't work that well. We found some interesting protective associations. As I mentioned at the beginning, that was kind of the thing that one of the things we're really interested in. The biggest and strongest hit that we have has sort of been suggested in the literature before, which is this protective PTV in IL-33 that decreases the risk of carriers for asthma. And we see this very high level of significance, like 10 to the minus 13 for the p-value. So I think there's sort of a preponderance of evidence in the literature now that suggests this is hopefully a real association. It might be interesting. We also, I didn't mention this, but among the phenotypes that we defined, we included 10 that were family history. So these are questionnaire questions, and instead of asking, do you have breast cancer, we looked at the answers to the questions: did your mother have breast cancer, or did your father, or did your mother or father have high blood pressure? And we pulled out a number of associations for family history. I think there were I have to remember, maybe 8 in total. But even for some of them, we actually see the same PTV associated both with family history and primary diagnosis. So for instance, there's a PTV in Paul B2 that's associated both with family history of breast cancer as well as sort of a primary diagnosis of breast cancer. So that's pretty nice. Once again, this is just the family history is relying entirely on questionnaire data. So it's nice to see this agreement between not only questionnaire data, but family history questionnaire data and sort of primary diagnosis from hospital records or also questionnaire data. And similarly, there's a PTV in FOOT2, which was associated with family history of high blood pressure and also with primary diagnosis of hypertension. Down on the plot, I've shown a few other ones that were somewhat interesting to us or maybe already known. So BRCA2 in lung cancers, there's known to be an association there. We don't see BRCA2 in breast cancer. And I assume the reason for that is the participants in the biobank are, I believe, at least 40 or maybe even 50. So we might be getting in a little too late to see a lot of the people that have the BRCA2 mutations. Maybe it's particularly females. That might be something that's a little more interesting to explore, but also something worth keeping in mind. The cohort here is an older cohort. We see a slight association between a PTV and VCORC1 and hypertension. And VCORC1 is one of the genes that's important for metabolizing warfarin. We're not sure if this is real. I'm still actually trying to see if I can kill this signal before we submit, but it's somewhat interesting. And also the NOL3 muscle or soft tissue injuries is kind of an interesting one. NOL3 is a gene that's expressed, in the muscle quite highly, and I think maybe has been implicated before, but it's pretty interesting. It's a pretty large effect for a PTV that I guess gives you a risk for having a muscle or soft tissue injury.
What would be the putative mechanism of loss of function VKORC1 that predisposes to or protects against hypertension?
Right. Yeah. So we don't know, and that's why we're kind of suspicious of that.
So you do know it's the effect size that you estimate from the data,
right?
But what's the odds ratio?
Oh, the odds ratio itself. Yeah, I can get the odds ratio. Let me look it up. So in this case, for VCORC1, the odds ratio is 0.97, and the confidence interval is 0.96 to 0.98. So it's a small.
So it's a small protective effect consistent with what you'd see in common variant association studies for common variants, right? So what you see in that cascade plot is that a lot of the common variants seem to have effects consistent with other common variant effects. Even though they are PTVs, this one seems to go in the protective direction, and you start seeing stronger effects as you go down in the frequency, and as you go to rare and lower frequency PTVs, which is what we've seen in other settings as well, with other PTVs that have been identified to be associated to phenotypes.
Is there any association with FOOT2 and stroke?
Great question. So FOOT2 is pretty pleiotropic. I think there was a GWAS. So did you show the plot already, Chris?
I'm going to get to it in just a second.
Yep.
Yeah. If we hold on that, I'll show you all the associations for FOOT2 in just a few slides. Great. Thanks for that, Manny. Yeah. So as Manny mentioned, this plot here, I should have introduced it to you guys. I apologize. So this is we call this a cascade plot. As you can sort of see the blue waterfall there. And basically what you're looking at on the x-axis is the minor allele frequency of the variant, and on the y-axis, the odds ratio, or the log odds on the left to the right. And you can see this sort of relationship between effect size and minor allele frequency that we've seen before. And certainly the VCORC1 variant is quite common. The minor allele frequency is 25%. I'm looking at my spreadsheet over here. And so, yeah, the size is pretty small, but for some of the more rare variants, the effect size starts to grow. Okay, so, and if we look back to the Manhattan plots, there was a lot of these sort of variants that are falling in the MHC region. So we decided to take a look and see whether or not these were truly independent PTV associations or whether they may be driven by HLA subtypes. So we did a conditional analysis for the 47 associations that were for PTVs in genes that were in or near the MHC. And on the right, what I'm showing is just the p-values conditional upon one of the HLA subtypes. So the UK Biobank provided HLA gene subtype information for each person, which was super useful. And you can see that most of these p-values drop down. So all the colored dots are the different PTV medical phenotype associations. So before, these were all quite significant, but then most of them sort of go back to not a significant association conditioned upon one of the HLA subtypes. And this kind of goes back to what I mentioned earlier, and there's a paper on the archive right now that's talking about this, basically saying that even for PTVs, which are predicted to have a large effect on genes and on coding sequence, they can still be in LD with something else that might be the sort of driver of the association. And in this case, I'm not sure whether there's still a few that have small p-values, but it's not really clear whether these are independent associations or not. We also took a look at the imputed data to see if we could pull out any more PTVs that we could also test for associations. And we were able to identify about 550 PTVs outside the MHC region among the imputed data. And we identified a handful of associations using the same sort of methodology that I just described in terms of binning them into different minor allele frequency bins. And we found some that are definitely known, so glaucoma and MyoC, or MAPT and Parkinson's disease. There's a couple others that I thought were kind of interesting, including this D2H-GDH and asthma and hay fever, which I think has been which was found in an earlier study of the initial UK Biobank release, as well as one of Joe Pickrell's papers in 2015, I think. So that looks to be somewhat interesting. We also found some associations between RPL3L and atrial fibrillation or flutter. Overall, the array contained a lot of PTVs, so we were looking at 18,000, so 546 didn't grab us too much more. I think that with better imputation, we'll be able to pick up some more, but it's nice to see that we're able to get at least a few from the imputed data. Okay, so that's basically everything for the GWAS. I'm gonna jump into the PheWAS and then this collapsed analysis, and these sections are shorter, so I won't be going too long. And the two figures still coming up, so that'll be here in a minute. So for the PheWAS, what we did is we really focused in on 25 genes. 17 of these come from the GWAS that I just showed you, and they were the genes that had at least one significant PTV association. We added in 8 more genes from the literature that were known to have, or that have been shown to have protective associations in a PTV in those genes, and you can see those in the table on the right. And here we've expanded the phenotypes beyond the initial 135 to include those with, to go actually to 206 to have at least 1,000 cases. So the idea here is, let's take a look at these genes which we've implicated or have been implicated in the literature as being important, and just get an idea about how they might affect other phenotypes. And we focused still in this case on PTVs with minor allele frequency greater than 0.01%. So of those 25 genes, 8 of them were associated under this sort of less stringent cutoff with 8 or more phenotypes. And there were several that had a handful of phenotypes they were associated with. And you can see all the different ones in the supplemental figure in the paper. I'm just going to go through one, which we put as a main figure that we thought was kind of interesting, which is IFIH1. So IFIH1 is an immune receptor that's sort of involved in detecting viral infections. It detects viral DNA and sets off the immune pathway to sort of fight a viral infection. And we see there are four.
The other thing to comment is that IFIH1, I think was the second published result where a PTV was shown to be protective against the disease. This was published, I think, around 2011 by John Todd and Serge Negentsev. I think, actually, it may have been 2009. And they showed a protective association to type 1 diabetes, which makes it a fantastic candidate for drug targeting. The other interesting tidbit was that while they were observing protection to type 1 diabetes, in our inflammatory bowel disease data set, we were starting to observe risk effects of those PTVs. And as Chris can show you, feel free to go through this slide, Chris. It seems to have quite a pleiotropic effects across other conditions.
Yeah, absolutely. So you can see on the slide here, we have these sort of 3 panels that are organized or sort of sorted vertically. So if you look across, maybe the top row, for instance, you see for the phenotype endometriosis, you can see on the left panel, we have the number of cases that we were able to find in the UK Biobank in the thousands. There's a minus log 10 p-value in the middle panel, and the different colors represent a few different PTVs that are in IFIH1. And then on the right, we have an estimate of the effect size of the association. I've grouped them on the left by phenotype. So you can see, for instance, in the middle there's hypothyroidism, and you can see that each of 3 PTVs sort of had, under this p-value cutoff, a significant association with hypothyroidism. And on the right, you can see that the odds ratios are all pretty much in agreement, the effect sizes. You can see similar things for psoriasis and ulcerative colitis at the bottom. And as Manny mentioned, there's protective effects, but there's also risk effects. So you can see the risk effects for IBD and ulcerative colitis that Manny mentioned, as well as some protective against things like psoriasis. So yeah, FUT2 seems to be involved in a variety of phenotypes. I'm sorry, IFIH1, sorry. And it's quite interesting, I think. Great, so as I mentioned, for the other genes, I'm not gonna go through them here one by one, but I'm glad to discuss any of them, and you can definitely check them out in the supplemental figures of the paper. Finally, I'm just going to go through this last analysis, this collapse and additivity analysis. So what we did here was, we asked for these same 25 genes that we were just talking about, the 17 that we've found in this paper, plus 8 from the literature. We wanted to look for associations between these medical phenotypes and the PTV carrier status for these genes. So basically, rather than looking at an individual PTV, we took each person and we asked, do they have one PTV in a gene? Then let's consider them a heterozygous knockout. So in this case, we're sort of assuming loss of function.
Yes, so just a correction. They're not, you can't, so it's not heterozygous knockout, because knockout implies having both genes down. So the way we could consider is one PTV is considered a knockdown and the 2 PTVs consistent with a knockout.
Sure, okay, sorry. Yeah, so heterozygous knockdown then. So let's say one functional copy, and then 2 PTVs would be no functional copies. And so then now we just have collapsed everybody down to like sort of a genotype per gene. And then we use these sort of carrier statuses to test for, in particular, departures from additivity. So, under the PLINK2 association analysis that we did, there's an assumption of additivity, which for these binary traits essentially comes down to assuming that whatever the log odds ratio is for the homozygote, well, it should be about 2 times the log odds ratio for the heterozygote. But under the non-additive model, we would see that these things are not equal. So there's a difference between what you get from having just one, the knockdown state versus the knockout state, as Manny mentioned. And so the most interesting of these, so of those 25 genes, we found evidence for sort of non-additive effects in 6 of them. But the most interesting comes from FUT2. And in particular, there's one PTV in FUT2 that's quite common in minor allele frequencies, like 49% in the UK Biobank. And FUT2 has been associated with a number of things, including Crohn's and psoriasis. And you can see down at the bottom here is a plot similar to the one I showed just a minute ago, but it's a bit different. So on the left, once again, we have sort of the phenotypes and the number of cases per phenotype. In the middle, we have the log 10 p-value, and on the right, we have the effect sizes. But now there's 3 points. The orange point are the values for the additive model. So you can see that, and this is why we identified FUT2 in the initial GWAS, which is that the additive model p-values are quite low, and there's an effect size. But if you split this up and you run a model that says, okay, let's separately test a sort of a knockdown or a dosage of 1 PTV or a knockout, a dosage of 2 PTVs, then you can see that over on the right or in the middle, for instance, the dosage of 1, there's really no significance on the p-value. And if you look at the odds ratios, they almost all overlap 1. Whereas if you actually have a true knockout of FUT2, then you have an increased risk for all these different phenotypes that we're looking at here. And I think there was a question of whether it was associated with stroke. I don't think we see an association with stroke here.
So you do see the association to hypertension. I think the stroke numbers in the biobank itself are still quite limited, but one thing that we can do is probably ask the collaborators from the stroke consortium to have a look at this as well in the meantime.
The numbers for stroke are pretty low.
So let me make a couple of comments.
Please.
You may or may not be aware of an association in the schizophrenia GWAS data with a gene called SLC39A8, which is a cation transporter whose major function is to transport manganese into the cell and into the Golgi. It turns out that manganese is the preferred metal by a lot for 2 sugar transferases, UDP fucosyl transferase and UDP galactosyl transferase. So there are 2 question comments. One is the UK Biobank have mental illness phenotypes to see whether FUT2 is associated with increased risk of schizophrenia, as is SLC39A8. And the second comment is, I wonder if it's worth a simple clinical trial feeding fucose, galactose, and uridine to patients with Crohn's to see if it improves them, because in a true loss of function of the manganese transporter, the sugar content, the glycoproteins in the blood, by measuring transferrin glycosylation, was abnormal, and although the patient was not improved clinically because it was a developmental problem, the blood glycosylation of the transferrin receptor was corrected by feeding the patient basically the precursors for the enzyme.
Those are all phenomenal ideas, Ed. I think it's those type of ideas started coming out once I started seeing these results as well. With SLC39A8, the story is the UK Biobank itself doesn't have enough schizophrenia patients. I think by combining verbal questionnaire data and hospital and patient record data, you probably get to around 800 to 1,000 cases. if you focus on hospital and patient record data alone, it's only like about 200 cases.
I see. I see.
So that's the opportunity that exists there, of course, is with combining data sets that exist. For example, that makes this at the Broad, combined with the data that exists in the UK Biobank. So there are some mental health level data sets available through UK Biobank. And one of the things that you start seeing with SLC 39A8, for example, if you look at fluid intelligence, is that carriers of that missense allele have lower fluid intelligence measures, and it's the top hit actually for missense alleles.
Really?
Yeah. So
what is fluid intelligence? I'm sorry, I'm not familiar with the term.
So I could send you the phenotype description that they've defined. I think Joel, I don't know if Joel Hirschhorn's on the call, but it's one of the measures that they have for mental health. And what I can do is to send you that link that describes the phenotype closer.
Terrific.
The other thing, as after this talk, what I'll do is to walk you through this biobank engine that would allow you to explore some of those questions as well.
Okay, that'd be great. Thank you.
And definitely the idea of the clinical trials for both SLC 39A8 and FUT2, given that they have that very simple management positioning, would be worthwhile to explore as well.
Okay, thank you. That is really an interesting association of the two genes, both that affect glycosyltransferase, a particular glycosyltransferase.
Absolutely, yeah. Thank you.
That can't be a coincidence in my biological perspective.
Yeah, and to follow up on what Manny said, we definitely don't have schizophrenia in this analysis. There's not even 1,000 cases, so it's something we'll have to look at more closely afterward, I think.
Yeah.
Yes, due to the sake of time, I would recommend we move forward, but we have another talk after this about the resource, the UK Biobank engine.
Yep. And this is basically it. I think so. I've kind of described this association and additivity analysis, and I will just give a quick overview of what we found. So this is sort of summarized in this figure here, which is, got the number of people that we've looked at. As I've talked about, we identified about 18,000 PTVs. And in the end, through the GWAS, we pulled out 28 associations outside of the MHC. And we took forward the genes that were implicated in these associations for both a PheWAS and a sort of knockout analysis. And, I've gone through some of the interesting examples. So we just talked about FUT2, and I showed IFIH1 earlier. We found some interesting protective associations, including strong evidence for this IL-33 and asthma association. And I'll just briefly say, so, the thing that we're still thinking about now is how do we get down into the lower minor allele frequency bin and really understand how get some power to detect associations for those really rare variants, whether they be PTVs or otherwise. And so that's stuff that we're working on now. And then I think as probably a lot of people appreciate, these PTVs in particular being rare, the thing that really determines their frequency is what population you're looking at. So for PCSK9, I've just got a little plot here from an app that we put together that kind of gives you an idea about what your power to detect an association, sort of the type that I just talked about, these sort of collapsed associations. So per gene, if you collapse the PTVs, what's your power to detect an association in different populations? And here I've got the UK Biobank and I've got different NOMAD populations. So for PCSK9, the PTVs and PCSK9 are a lot more prevalent in African population than the other ones. And this is true if you look at different genes, and I encourage people to check out on the biobank engine. You can throw in different genes and see how it changes. And so, it's going to be important to take a broad view of different populations to really take advantage of this sort of natural experiment of PTVs and understand how they affect genes, or how they affect a broad range of phenotypes. So I'll leave off there, and I'll let Manny take over, and I'm glad to answer any questions later on or via Twitter or anything else. Thank you.
Thank you very much, Chris. It was very great. Due to the sake of time, I would recommend that we directly move forward with Manny.